So this is Beth. She also started with Scott at the same time I did. She is talking about something a little different, but she's going to Cornell next year to pursue her doctorate in natural biology. But yeah, here's Beth.
You do be letting anyone in now. All right. In the beginning, we haven't even gotten to the fun part. So, like Lindsay mentioned, my name is Beth. My thesis project is titled Towards an Electrophilic Inhibitor of Bacterial Quorum Sensing Synthase TOF-I. So, long story short, not cancer. So, there's a few things we're going to cover in this presentation, but the first thing is most pertinent is an overview of what quorum sensing is. So, quorum sensing is a method of cell-to-cell communication utilized by bacteria to coordinate population size with gene expression. In essence, it's a way of bacteria to speak and listen to one another in order to engage in group behaviors cooperatively. They do this by means of a small molecule called an autoinducer, which I illustrated as a little yellow bubble. You can think of this like a word that is spoken by bacteria and listened to by bacteria. There are two main proteins in this quorum sensing system, the first of which is the autoinducer synthase, shown here in pink, which, as the name suggests, synthesizes the autoinducer. Once this happens, the autoinducer is able to diffuse freely through the bacterial membrane into the extracellular environment. Now, as the population of bacteria in a given area increases, so does the concentration of autoinducer in the extracellular environment. Eventually, it reaches a point where the autoinducer is able to diffuse back through the bacterial membrane and bind the second protein in the system, the autoinducer receptor. When this happens, the receptor undergoes a conformational change, which allows it to bind DNA and activate the transcription of target genes. So this is how bacteria speak with the synthase and listen with the receptor. But the main question is, why are we so interested in how bacteria talk to each other? Well, it turns out that the quorum sensing system has been implicated in pathogenicity and virulence factors in a variety of human and plant pathogens. So by stopping this non-essential communication pathway, it presents a novel opportunity of designing anti-virulence compounds that don't impose selective pressure because they don't kill the bacteria. In reality, though, gram-negative bacteria's autoinducers do not look like little yellow bubbles. They look like this. The conserved structure among gram-negative bacteria is called an acylated homoserine lactone, shown here, which is conserved among species. And the main differentiation occurs in oxidation at the C3 position, so the presence or absence of that carbonyl group, and acyl tail length. Like I mentioned, the quorum sensing system is a drug target, and since there are two main proteins involved in the system, there are two main methods of inhibition. The first of which, shown in panel A, is receptor inhibition. So if I draw your attention to the extracellular environment, you'll notice that there's not only just little yellow bubble autoinducers, there's also a purple bubble, which is the receptor antagonist. The receptor antagonist outcompetes the autoinducer to bind and inhibit the autoinducer receptor, thus preventing the transcription of target genes. The autoinducer synthase, however, is unaffected. It's still able to produce autoinducer, but the presence of the antagonist kind of outcompetes it. This creates a bacteria that is functionally deaf. It can still speak by producing its own autoinducer, but because the antagonist is bound to the receptor, it can no longer hear. So this compound is like the bacterial equivalent of earmuffs. If we look at panel B, this is the second mechanism of inhibition. If you notice in the extracellular environment, there's no more autoinducer. This is because there's these gray triangles, the synthase inhibitor, that blocks the formation of autoinducer by inhibiting the synthase. You'll notice that the receptor remains unaffected, but you still get the same response, no transcription of target genes. Bacteria is no longer able to speak, and just because it can still listen, there's nothing for it to hear, so you don't get the same response. So this creates a bacteria that is functionally mute. It can still listen for things in the environment, but it can no longer speak by itself. So if you want to think of this compound like putting duct tape over its mouth. Of these two mechanisms of inhibition, we're more interested in synthase inhibition for the reason that there are only two published synthase inhibitors. There are literally thousands of receptor antagonists, and this has been developed as a common drug target in prior research, but synthase inhibition is largely undeveloped. This structure down here is known as J8C8. It's the only other published inhibitor of an autoinducer synthase, and this is its crystal structure right here called TOF-I. TOF-I is a synthase that J8C8 kind of antagonizes, but this is the only other known synthase inhibitor besides ours. So now this is how we inhibit the synthase, but how does the synthase actually work? How do we design these compounds? So our synthase of interest is SEP-I. It's from the bacteria Burkholderia cenosepacea, which I believe is a human pathogen. And SEP-I synthesizes the C8 autoinducer. And C8 just refers to the number of carbons in the acyl tail here, shown in green. This autoinducer is synthesized by a combination of S-adenosylmethionine, or SAM, and acylated acyl carrier protein, acyl ACP. First step of the mechanism is just a substitution reaction in order to form this amide bond and kick off the IOACP. And then there's this lactinization step, the transition state, which separates the autoinducer from methylthioadenosine, or MTA, the side product. So we wanted to take a closer look at the transition state, this lactinization step. It's been shown in enzymatic studies that in order to design an inhibitor, you kind of want to make one that looks like the transition state because that's when the enzyme is bound most tightly to its two substrates in order to catalyze the reaction and make them in the proper orientation. So if we look at the transition state, we look at the transition state structure shown here, and we look at our first synthesized inhibitor, JKR1, named after a prior student, Julian Kellner-Rogers, you'll notice remarkable similarities because they include both the lactone moiety and the adenosyl moiety here. So it matches the transition state, which is why in this data, this dose response curve, you see as the concentration of JKR1 increases, the activity of the enzyme decreases. So this is kind of proof that this design method does work. But why stop here? This is where I kind of stepped into the picture. Over the summer, about 2 years ago, I gained this mania for covalent inhibition. And so I decided, what if we make this a covalent inhibitor? So structural analysis of TOFI, the enzyme inhibited by J8C8, has shown the presence of a cysteine residue way back in the acyl tail binding pocket. By sequence analysis, this cysteine residue is also present in CEPI, our enzyme of interest. So my whole idea is in this big box here. What if we change the terminal methyl group on JKR1 to an electrophile in order to bind this cysteine? This has been done in the past in prior drugs, so why can't we try it as well to develop the first covalent inhibitor of any quorum sensing enzyme? The three electrophiles that I wanted to focus on were first this bromine atom, then an acrylamide, and an isothiocyanate. Now, I'm allowed to have a favorite electrophile because naturally, so my favorite electrophile is the acrylamide, and mainly because it has been shown in prior pharmaceuticals to be an effective inhibitor of cysteine residues. So this is osimertinib, an EGFR inhibitor used in non-small cell lung cancer, and this is sotorasib, which obviously I have to talk about, a KRAS G12C inhibitor. And these acrylamides are shown in both compounds because they both target cysteines on their respective proteins. So this is a known and shown method that I wanted to test out as well. So that's the idea. This is how we're going to make them. So my first target was the synthesis of bromo-JKR shown here. It's probably the easiest one to kind of functionalize. And so this is what we're trying to do. We're starting with two separate reactions that we're going to bring up these starting materials and then combine them together. So up top, we're starting with allyl glycine, using a Bach group to protect the amine, and then using NBS and THF to do this bromo-lactinization reaction. It's pretty cool. And on the bottom, simultaneously, we're bringing up our thioadenosine. So we're starting with isopropyridine adenosine, using thioacetic acid to do a Mitsunobu reaction, and now we have both of these products to begin with. We're going to combine them together with sodium methoxide and methanol in order to make this bond. So it's starting to resemble our final structure. The last thing we have to do is add the acyl tail. The acyl tail is added by deprotecting the Bach group with trifluoroacetic acid, and then adding on this bromo-octanoyl chloride with TEA to make this other amide bond shown here. And then we just remove the acetal protecting group on the bottom of the isopropyridine adenosine with the 1-to-1 mixture of formic acid and water. One thing I wanted to note about the strategy of the synthesis is our electrophile is a bromine. We use a bromine as a leaving group here. So we can't add the acyl tail before we do this coupling step because there's kind of no way to determine where the reaction is going to happen. So we had to push this off until the end. So one electrophile synthesized. Now it's time to move on to the two others we proposed. Next is the synthesis of the isothiocyanate JKR, or ITC. So this is our target right here. A very similar beginning process. We make that Bach bromolactone, but this time we actually deprotect it first because we're simultaneously bringing up this azide, like acid chloride, because we're going to add the acyl tail on in the beginning, since this azide isn't going to pose any problems for the remainder of the synthesis. We're going to couple those together with DIPEA to form the amide bond shown here. Once again, Mitsunobu reaction to make our thioadenosine, couple them together, and now we're here. We're 2 steps away from the finish. We need to do a Staudinger reaction to convert the nitrile, the azide, into an isothiocyanate, and then deprotect the acetal on the bottom. But because of the low yields of the prior reactions, we didn't have enough of this compound in order to continue with the Staudinger condition, so we kind of stopped here just as the azide, which is a non-electrophilic JKR. So in future, if we wanted to run the synthesis again, we're going to have to up the ante on it. So with that in mind, we're going to start to the final electrophile, the acrylamide shown here. Very similar beginning steps. We make the Bach bromolactone, But in this case, instead of using our regular acyl tail, we're using Bach GABA shown here. So we're using DCC to make the amide bond as coupling reaction. Same Mitsunova reaction, and then stick them together with sodium methoxide and methanol, or so we thought. When we took an NMR of this compound, the Bach group just disappeared. So we're not entirely sure what we made, but it wasn't this because the Bach group is usually pretty easy to characterize on an NMR spectrum. So if that's not there, we're not entirely sure what this reaction yielded. Regardless, we have one electrophile made, and now comes the fun part, we have to figure out how to test it. So I mentioned earlier that J8C8 is an antagonist of TOFI, and TOFI is a related synthase to our enzyme SEPI. They catalyze the exact same reaction to form the exact same C8 acylated homoserine lactone autoinducer. When we figured this out, we thought, why not have both of these enzymes kind of up and running in order to do further testing? So over the summer, I purified TOFI, and now we have 2 enzymes to test against instead of 1. The first thing we did when we got our TOFI purified was ensure that JKR1 was still a reversible inhibitor of both enzymes. Since they catalyze the exact same reaction to form the exact same product, they should have the exact same transition state, and so our transition state mimic inhibitor should function on both. This data confirms that because as the concentration of JKR1 increases against both enzymes, the activity decreases. So knowing this going forward, this was a good kind of encouraging data to have moving on to testing the electrophilic inhibitors. The next thing we did was test bromo-JKR1, the only electrophilic inhibitor we successfully synthesized. So once we did this, we tested it against TOFI, and we got this dose response curve. This was incredibly encouraging because we changed the methyl group to a bromine atom, so there is a possibility that the compound might not even access the binding pocket because of the difference in structure. So knowing that bromo-JKR1 can function as a reversible inhibitor was a key first step before determining if a covalent bond was formed. Our next assay was to determine if this covalent bond was formed. This was done by what's called a jump dilution assay. So jump dilution assay is done in a 96-well plate, or for my purposes, a 4-well plate. We're starting with a concentration of bromo-JKR1 well above its IC50. So here it's at 10 micromolar, and we're incubating it with TOFI. What we're going to do next is we're going to dilute that mixture 30-fold into the full assay contents. So this is going to effectively wash the concentration of the drug down to a concentration below its IC50, where we should not expect to see inhibition. So in the dark red, we should expect to see inhibition. In the light pink, once it's in the full assay mixture, we should not. If our compound is covalent, these are the results we expect to see. So JKR1 is kind of our control. It does not matter how much time it spends incubating with TOFI, it is not going to form a covalent bond. It is a non-electrophilic reversible inhibitor. So no matter how long we let it sit with TOFI, be it 0 minutes, so immediately dilute into the assay, put it in the instrument, run it, or let it sit for 3 hours and then dilute and run it, we should not see any difference in inhibition because it's still functioning at a 0.33 micromolar concentration. The difference comes with bromo-JKR1. If it forms a covalent bonds take time. So if we immediately dilute our enzyme and drug combination into the assay mixture, we haven't given it enough time for a covalent bond to form. So we should see no inhibition. But after 3 hours of incubation time, that should be plenty of time for a bond to form between the synthase and the compound. This means that when we do the dilution, it shouldn't matter because they're permanently linked together. So we should still see inhibition even at that sub-IC50 concentration. So keep in mind, this is what we expected to see. This is our data. Let's walk through it a bit. No enzyme, we shouldn't see any activity. Here's full enzyme TOFI activity. JKR1 was our control shown in blue. We shouldn't see any inhibition at either time period. Bromo-JKR1 shows no inhibition at either time period. This indicates that a covalent bond was not formed. Because there is no inhibition shown here, this bar should have dropped down if we had a covalent bond formed, so we did not form a bond between bromo-JKR1 and TOFI. So kind of to wrap up the past few years of my time in the lab, 3 things have happened. New molecule, new enzyme, it works. So we synthesized 1 successful electrophile bromo-JKR1, and we purified TOFI, the related enzyme, to SEPI, and we confirmed that this electrophilic derivative of JKR1 still maintains some reversible inhibition against the enzyme. This is kind of key moving forward if we're going to keep making changes to the end of the acyl tail. Some future directions would be to switch to J8C8, that other known inhibitor of a quorum sensing synthase. You don't have to be an organic chemist to kind of see that this looks a lot easier to make than JKR1. The synthesis is quite simple, and that means it's easier to functionalize at this end position in order to add electrophiles. So the flash to bang from synthesis to testing isn't going to be as long. So we're able to make more derivatives of this compound and determine if a covalent bond can even be formed in the first place, if that cysteine is even going to be reactive if we put an electrophile near it. So future students in the lab might have a lot less time synthesizing things. So lastly, I'm gonna hit the brakes on the science for a bit and embarrass a few people in this room. So on this slide are some of my closest friends and best mentors. Honestly, it's been a pleasure being able to interact with these folks over the past few years. And I could stand up here for like 20 minutes and talk about it, but I'll spare the details. The only other thing I wanted to mention is that 3 years ago, I sat 3 rows back and 2 seats over from 9:25 to 10:40 a.m. on Tuesdays and Thursdays. And that's kind of where everything started for me. And I don't know where I would be if it weren't for that class and the person who taught it, who I'm trying not to make eye contact with. So that's the reason I wanted to do work in organic synthesis. Is the reason I'm pursuing a PhD in chemistry. The reason I'm standing up here. So kind of a good full circle moment to be standing in the same spot. Thank you all again for a great, pandemic aside, strange 4 years. And I wouldn't have changed any of it for the world. So with that, any questions? Yes.
As to, you're looking at, there's just these 2 synthase inhibitors. Do you think that there are so few of them simply because people haven't been looking for compounds that work in this way? Or is there something biochemical there that would explain why there's only a limited number of things that'll work?
I'm not entirely sure. I can say from like the literature I've read in background, kind of the first inhibitor was a receptor inhibitor. And so that's kind of where everyone followed onto that train. And so when J8C8 was published, I guess it didn't get a ton of like hype. And so everyone's been so focused on receptor inhibition instead of synthase inhibition. So I'm not entirely sure why everyone kind of bandwagoned the receptor, but there are 2 published synthase inhibitors.
There might be a huge number of.
There might be like plenty more that just have yet to be kind of figured out.
Are you concerned when you add your electrophilic group that it will change the permeability of that membrane and it won't be able to access?
Yes, in future, yes. So we're not doing any cell-based assays right now. It's just like purified enzyme. And so once we get to a point where this could be a more drug-like compound, right now, we look back at what JKR looks like. This is not a very good drug-like compound. It's huge and polar. So we'd have to do some more tinkering with it in order to talk about like permeability assays. Yes.
I just wonder if there's something magical about 180 minutes. Like why not 5 hours?
You know, there's not really anything magical. So when Scott and I were trying to figure out how to design assays to test for covalent inhibition, which I don't think either of us really knew how to do, we kind of followed a protocol. It said 3 hours. We're like, okay, 3 hours. Yes.
There are also photoinducible ones. If you're working on this ex-vivo system, you can put on a photoinducible covalent so that the factor is like that something can happen.
Like a photoprocessor.